Okay, so today we are going to be reviewing prokaryotes. If you haven't already seen our SI folder on iLearn, we created a worksheet for this topic, and this video should hopefully help you out with that. So let's begin. We have three main domains: bacteria, eukarya, and archaea. Prokaryotes are comprised of bacteria and archaea. If we compare prokaryotes to eukaryotes, prokaryotes are much smaller in size than eukaryotes. Prokaryotic cells have a cell membrane but no internal membrane-bound organelles. But the cell membrane can be folded to make special areas for enzymatic function. Prokaryotes do not have a nucleus, and the genetic material is free-floating in the cytoplasm. Prokaryotes have a small genome in comparison to eukaryotes and do not have any organelles besides ribosomes. Ribosomes do not have a cell membrane, which relates to my earlier point that prokaryotes do not have any membrane-bound organelles. Here we have a prokaryotic cell. Prokaryotic cells contain free-floating DNA, which can be referred to as a nucleoid that contains the genetic information. There is the plasma membrane, which helps the cell maintain its shape. As we talked about earlier, you can see that the plasma membrane is making some infolds. Next, we have the capsule, which is the sticky protective layer used for adhesion. Prokaryotes have ribosomes, which are the protein factories. Note that ribosomes are the only organelles that the cell has. There's also the flagella that helps in cell movement and locomotion. Lastly, we come to the cell wall. The cell wall maintains cell shape and protects the cell. I saved the cell wall for last because there is a significant difference in the cell walls of archaea and bacteria. Bacteria and archaea are both prokaryotes, but bacteria contains a unique molecule in its cell wall called peptidoglycan. The composition of the cell wall and the location of peptidoglycan within the cell wall determines whether the bacteria cell is gram-positive or gram-negative. In gram-positive bacteria, a thick layer of peptidoglycan makes up the cell wall. That's why, when stained with the gram stain, the cell glows bright purple. In gram-negative bacteria, peptidoglycan is sandwiched between two layers of plasma membrane. The stain cannot easily access the peptidoglycan, which causes it to glow pink. Just remember that this is unique to bacteria. All prokaryotes reproduce via a process called binary fission. In binary fission, the chromosome undergoes replication into two bacterial chromosomes. When the cell grows, they separate as plasma membrane is inserted between them. The cell expands and is pinched into two daughter cells that each contain one chromosome. Binary fission is a very fast process, which means that there is a lot of room for mistakes. In biology, we refer to these mistakes as mutations that contribute to prokaryotic diversity. Another factor that contributes to prokaryotic diversity is genetic recombination. There are three main types of genetic recombination. We have transformation, in which a cell can absorb and incorporate foreign DNA from external surroundings. DNA may be then integrated into the genome or exist as a plasmid. We also have transduction, which is the movement of genes between prokaryotes by viruses. Sometimes during an infection, a virus will package bacterial DNA into new viruses. The new viruses donate bacterial DNA into a new host, and if the new host cell survives, it maintains the recombined DNA and now contains DNA from a previous victim. Lastly, we have conjugation. A cell that exhibits an F factor gene can initiate conjugation, in which a sex pili harpoons a recipient cell and forms a mating bridge that allows the DNA transfer process, which includes replication of DNA. The recipient cell inherits the F factor gene and the process repeats. One last topic I want to cover is the nutritional and metabolic adaptations of prokaryotes. Photoautotrophs use light as their energy source and CO2 as their carbon source. Chemoautotrophs use inorganic compounds as their energy source and carbon dioxide as their carbon source. Photoheterotrophs use light as their energy source and organic compounds as their carbon source. Chemoheterotrophs use organic compounds as both their energy and carbon sources. There is also some other vocabulary that you should be aware of. Photoautotrophs and chemoautotrophs are both autotrophs because they use CO2 to make compounds. Photoheterotrophs and chemoheterotrophs are both heterotrophs because they use organic nutrients to make compounds. Photoautotrophs and photoheterotrophs are both phototrophs because they get their energy from light. In contrast, chemoautotrophs and chemoheterotrophs are both chemotrophs because they get their energy from chemicals. It's a lot of vocabulary, so just make sure you are comfortable with it. That's all for this video. We hope it was helpful, and we will see you during office hours. Thanks for watching.